Hello guys, my name is Dhanasri and I am from ISMP Science.
My name is Mirasri Doitong My number metric is D20181083374.
Assalamualaikum dan salam sejahtera. Nama saya Siti Nurhaliza binti Abdul Aziz Muhammad Syed di 20181083387 daripada ISMP Science. Assalamualaikum dan hai, nama saya
Hello guys, my name is Dhanasri and today I will explain about animal evolution. Animal evolution is a change in a gene pool of a population from generation to generation, such as mutation, genetic drift, and natural selection.
DNA really make mistake, but can happen. A point mutation change a single base. It can change the whole sequence of amino acid, while deletion remove a base and cause frame shift. It can change every amino acid after the mutation. Originally, the codon will be form pattern, but after the deletion, the amino acid sequence will change. The result will be non-functional. While insertion add a base and cause frame shift. Originally, codon will be form pattern, but after the insertion, the amino acid will change because base shift by 1. While inversion invert a segment of DNA end to end. Each bases removed around. The start codon begin to coding the protein. If the mutation happened before or after the protein coding sequence, there will be non-effect on protein. Mutation must occur in the coding section of DNA to affect the protein synthesis.
We are going to present about mechanism of evolution. One of the first mechanism of evolution is all in gene and DNA.
If Darwin came back today, what will he be most excited by in a modern science? I think it will be the whole world of genetics. That is the answer to all the big questions he had. How does variation emerge? And how can that be transmitted? Let's return to our brown bear friend in Arctic to consider the impact of genetics on our understanding of evolution. Each bear is made up of cells, and if we take brown bear cell and tunnel into its nucleus, we find DNA, the molecule with genetic instruction for building, in this case a brown bear written letter code. Now the thing about DNA, it is not perfect. When it copied, mistake get made. Mutation, in other words, that sometimes affect an organism trait and that sometimes can be passed from parents to offspring. So the variation at the end of evolution, it is genetic variation. Slight difference in DNA that, for example, could make some bears a little light in color, a bit more insulated against the cold, and a bit more capable of digesting fattier food like seals. Evolution is essentially any changes in the genetic composition of a population. Mutations are random, so they are not always helpful. But bears with mutation that give them some advantage for Arctic living survive and reproduce more often than bears without them. They pass the gene responsible for those adaptations onto their cubs. Over generation, more bears inherited and elaborated on this and other changes in the DNA. The eventual result, polar bear. And when tunnel into its nucleus, we find a polar bear DNA. Think of DNA as a raw material that across billions of years evolution has modeled and built into countless forms of life. We are all related tied to one another by the most spectacular of double helical thread. Macroevolution is the large-scale pattern, trend, and rate of change among families and other more inclusive groups of species. This is the example of macroevolution. Microevolution is a change in allele frequency resulting from natural selection, genetic drift, gene flow, and mutation. The Darwin finches is example of the microevolution. Natural selection. Natural selection refers to differential survival and reproduction among individuals. Divided into three: directional, stabilizing, and destructive. Directional is a shift in the average value of a trait to one direction only over time. Stabilizing is a genetic drift decreases as the population stabilizes on a particular trait value. Destructive is selection for extreme trait values and result in two different values with selection against the average value. This is example of directional selection, stabilizing selection, and destructive selection.
Allopatric speciation occurs when a species separates into two separate groups which are isolated from one another. A physical barrier such as a mountain range or a waterway make it impossible for them to breed with one another. Each species develops differently based on the demands of their unique habitat or the genetic characteristic of the group that are passed on to offspring. When Arizona's Grand Canyon formed, squirrel and other small mammals that had once been part of a single population could no longer contact and reproduce with each other across this new geographic barrier. Today, two separate squirrel species inhabit the north and south rims of the canyon. On the other hand, birds or other species that could easily cross this barrier continued to interbreed and were not divided into separate populations. Parapatric speciation sometimes happen when part of an environment has been polluted. Mining activities leave waste with high amounts of metals like lead and zinc. these metals are absorbed into the soil, preventing most plants from growing. Some grass, such as buffalo grass, can tolerate the metals. Buffalo grass has become a unique species from the grass that grow in areas not polluted by metals. Long distance can make it impractical to travel to reproduce with other members of the species. Buffalo grass seeds pass on the characteristics of the members in that region to offspring. Sometimes, a species that is formed by parapatric speciation is especially suited to survive in a different kind of environment than the original species. Example of sympatric speciation is the apple maggot, an insect that lays its eggs inside the fruit of an apple, causing it to root. As the apple falls from the tree, the maggots dig in the ground before emerging as flies several months later. The apple maggot originally laid its eggs in the fruit of a relative of the apple, a fruit called a hawthorn. The original hawthorn species still only lays its eggs in hawthorns. When small groups of individuals break off from the larger group and form a new species, this is called peripatric speciation. As in allopatric speciation, physical barriers make it impossible for members of the groups to interbreed with one another. The main difference between allopatric speciation and peripatric speciation is that in peripatric speciation, one group is much smaller than the other. Unique characteristics of the smaller groups are passed on to future generations of the group, making those traits most common among the group and distinguish it from the others.